Hello everyone, this lecture video is all about glycolysis. We can define glycolysis as a metabolic pathway by which the glucose, which is a 6-carbon molecule, is converted into 2 molecules of pyruvate, which is a 3-carbon molecule, wherein a chemical energy in the form of ATP is produced, and the reduced nicotinamide adenine dinucleotide, which is a reduced coenzymes, are also produced. This is a linear cycle instead of cyclic cycle because it does not regenerate the reactant, and it takes place in the cytosol. So here is the cytosol in case you are confused between cytosol and cytoplasm. Here is the cytoplasm. This light green here, it can be defined as cytoplasm is a cell component present inside the cell membrane, while the cytosol is a fluid or liquid fluid present in the cell membrane. So this is the fluid, the darker green here, the apple green in color. So the cytosol contains the enzymes, the organelles that is suspended, and some cell structures that is present in the cell. So this cytosol is where some of the enzymes that catalyzes the glycolysis occur. That is why the glycolysis takes place in cytosols, because the enzymes to catalyze the reaction are all present here. And then the word glycolysis originate from the Greek words glyco, which means sweet, and lysis, which means breakdown. This is also known as Embden-Meyerhof pathway, named after the German chemist or biochemist Gustav Embden and Otto Meyerhof, who discovered the pathway in 1930s. Then this is the overall overview of the glycolysis. Let us take that one by one, step by step. Here is the step 1, wherein the glucose will be converted into glucose 6-phosphate. So this one. Here we will be using an ATP for this reaction to take place. Let me see. For step 1, that is the phosphorylation using ATP. Once the monosaccharides, glucose, fructose, and galactose entered the bloodstream through the villi, in which you have watched in the previous lecture, the fructose and galactose passes on the liver, okay, which will be converted later to glucose 1-phosphate. So this glucose here will proceed to cytosol of the cells to produce a pyruvate. So here is your glucose from the bloodstream that entered the cytosol. So with the help of the enzyme hexokinase here and the utilization of ATP, we will be using an ATP here. We will get 1-phosphate. So I highlighted here the place here, the hydrogen here. This is where the phosphate will attach itself. When you say hexokinase, it is the rearrangement. Okay, the enzyme that catalyzes the transfer of phosphoryl group from ATP to a substrate. So when we say substrate, this is your substrate here, the reactant here, and then an ATP will be attached here, since it is the function of hexokinase. Hexo means it will replace in the 6th carbon. So the phosphate will replace the hydrogen here in the 6th carbon. Okay, this is what will happen. And the ATP, you will now have just the ADP because you reduced 1-phosphate. So you will now have, from triphosphate, you will have now the diphosphate. That is why this is named as glucose 6-phosphate. This is glucose, and the phosphate was attached on the 6th carbon here. Okay, so next, phosphorylation, meaning addition of phosphate in a glucose using an ATP. So here we used an energy. We did not produce an energy. We used or utilized an energy from ATP. Now, for step 2, isomerization. Isomerization is the rearrangement of the molecule. So you have here your glucose 6-phosphate, and with the help of the enzyme phosphoglucoisomerase, it will isomerize this one into 5-ring carbon. So we have here 1, 2, 3. So from here, you will have an ethylene glycol to compress this area. It will be compressed. So you will have now your oxygen at the top and an ethylene glycol here. This will be your 1, 2, 3, 4, 5, and then the 6th here. So this is now the fructose 6-phosphate, because again your phosphate is here at the 6 carbon, and since you arrange that into a 5-ring member system, that will be a fructose 6-phosphate. Next step is another usage or utilization of ATP. We are using an energy and we are not able to produce the energy here. So, phosphorylation using ATP, we will add phosphate on the fructose 6-phosphate. I outlined a green here. We will be using an ATP and one phosphate from ATP. So, you will now have the diphosphate ADP, and with the help of the enzyme phosphofructokinase, meaning to say we will attach an phosphate here, a phosphoryl group, and we will attach that here, and the phosphofructokinase will attach that on the 6-ring carbon here. That is why you have your fructose 1,6 biphosphate. You have your phosphate at 1 and another phosphate at 6. That is why you have now your fructose 1,6 biphosphate. So the use of biphosphate instead of diphosphate is to indicate that the two phosphates are on different carbons and not connected to each other. Unlike if we use the diphosphate, it means that they are in the same carbon. So for example, if you use diphosphate here, means to say there are two phosphates here. But since they are of the different ring system or of different carbons, we are using the biphosphate, indicating that there are two phosphates but of different places. Okay, next we have here step 4, the cleavage. When we say cleavage, that is the breakage of a, or splitting into half of a molecule. So this is your fructose 1,6-phosphate. This is an open chain form. For you to clearly see how the cleavage or breakage will occur. So this will be splitted into 3 carbon ring system into halves here. So, with the help of the enzyme aldolase to initiate the cleavage formation. And after the splitting of that, you will now have the dihydroxyacetone phosphate and glyceraldehyde. So, what will happen next? The dihydroxyacetone, we have to convert this into glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate will now undergo into the step 6 rather. So, the hydroxyacetone phosphate here, we have to proceed to step 5. So, step 5 is all about isomerization, since we need to convert this into the dihydroxyacetone phosphate, since the glycolysis requires a glyceraldehyde 3-phosphate. So, the dihydroxyacetone phosphate here, with the help of the enzyme triose phosphate isomerase, this enzyme isomerizes the placement of the bond and the hydroxyl group here. So, if you will notice here, I have here a double bond here and hydroxyl. They will switch their places to isomerize the glyceraldehyde phosphate here, the image of that, this one. Okay, they are just inverted. And then, you will now have the glyceraldehyde 3-phosphate. Next, for step 6, oxidation and phosphorylation using an inorganic phosphate. This inorganic phosphate, the I here is a subscript, so that is P sub I. Okay, so the glyceraldehyde 3-phosphate from step 4 will also undergo this process, and this one. So, the glyceraldehyde 3-phosphate here, since this dihydroxyacetone phosphate undergoes into step 5, right, to be converted into glyceraldehyde phosphate. So, there are 2 glyceraldehyde 3-phosphate that will undergo in step 6. From step 4, this one, and from the converted or the isomerized glyceraldehyde 3-phosphate here. So, there are 2 glyceraldehyde 3-phosphate that will undergo the steps 6 to 10. Okay. I want to emphasize that. So, the 2-glyceraldehyde 3-phosphate, 2 pieces of that, will react with the oxidized form of nicotinamide adenine dinucleotide plus an inorganic phosphate with the help of the enzyme glyceraldehyde 3-phosphate dehydrogenase. It will remove a hydrogen from here. Okay, and the hydrogen here will be attached on the oxidized form of NAD here. And the inorganic phosphate will attach on the place of the hydrogen that has been removed. So you will now have here your phosphate, and the hydrogen from here will be served as a reduced form of nicotinamide adenine dinucleotide, and hydrogen ion will be produced because this one is a reduced form, so mean to say it lost an electron. So from glycerol triphosphate, you now have your 1,3 biphosphoglycerate because you have your phosphate here in 1 and 3 carbons. That is why that is 1,3 biphosphoglycerate. Again, the use of bi to indicate that the phosphates are placed in different carbons. Okay, next, step 7. Now you have produced here 2 pieces of this because you have 2 pieces of glyceraldehyde triphosphate from step 4 and step 5. So you will have 2 pieces of 1,3 biphosphoglycerate that will undergo the step 7. So step 7 here, you have 2 pieces of 1,3 biphosphoglycerate, and we will be producing an energy here. So from here, we will be utilizing the adenosine diphosphate, and with the help of the enzyme phosphoglycerokinase, this phosphate here will be attached on the ADP here, and you will be left out with here with the oxygen. And the phosphate will be added on the diphosphate, and you will have the triphosphate. Therefore, you produce an energy. So the oxygen that is negative here, you will have that one. So you now only have the 3-phosphoglycerate because the phosphate here has been removed on the first carbon. You only have now a phosphate on the third carbon. So you produce 2 pieces of 3-phosphoglycerate because you have here 2 pieces of 1,3 biphosphoglycerate. So these 2 pieces of 3-phosphoglycerate will undergo the reaction in step 8, the isomerization. This isomerization with the help of the enzyme phosphoglyceromutase. The purpose of mutase, this is an enzyme that affects the shift of functional group from one position to another within a molecule. So there will be a shifting of functional group. So these 3-phosphoglycerate, the phosphate here will be isomerized or will be placed in or will be shift in position into the second carbon, since this is in the third carbon. So that will be placed in the second carbon, and the hydrogen here will be attached here. So you will now have here an ethylene glycol, and phosphate will be attached on the second carbon along with the oxygen. So this is now the 2-phosphoglycerate because the phosphate is now on the second carbon. And then you will produce here 2-phosphoglycerate because you have 2 pieces of 3-phosphoglycerate. So for step 9, the 2 pieces of 2-phosphoglycerate will undergo an alcohol dehydration, meaning to say we will remove a water, dehydrate. Okay, we will be removing a water with the help of the enzyme enolase. So from the water molecule that is attached on the second and third carbon will be removed. Therefore, you will produce a water, H2O, and the name of this enzyme is now phosphenol pyruvate. So you produce 2-phosphenol pyruvate because you have 2 pieces of 2-phosphoglycerate. So next, step 10 is the phosphorylation of ADP. So this is the second time that we are going to add phosphate on ADP, and therefore this is the second time that we will be producing an energy. So with the help of the enzyme pyruvate kinase, meaning to say the phosphate here will be removed and will be added on the ADP here to produce an energy, which is the ATP. Okay, this ATP is the energy that is being utilized by our cell. So we have 2 pieces of phosphenol pyruvate, right? And with the help of this enzyme will remove the phosphate here to be attached to the ADP, and you will now have the ATP, and this will be arranged into a double bond, and you will have a CH3 at the bottom. So this is now the pyruvate. Therefore, you produce again 2 pieces of pyruvate. So the definition of glycolysis is you will produce from 1 glucose, you will produce 2 pieces of pyruvate here. 2 molecules of pyruvate from 1 glucose. Sige. Let us count the ATP production and consumption during glycolysis. So these are the steps, the reaction, and the ATP change per glucose. So for step 1 and step 3, okay, for step 1, the reaction of glucose to glucose 6-phosphate, we utilize an ATP. So the ATP has been converted to ATP because we use an energy to convert the glucose into glucose 6-phosphate. And for step 3 as well, fructose 6-phosphate that is converted to fructose 1,6 biphosphate, we also utilize an energy. If you will look back here, this one, step 1, the glucose to glucose 6-phosphate, and then the fructose 6-phosphate, we also use an ATP to add phosphate on fructose 6-phosphate. That is why you have 2 phosphate here in first carbon and in 6 carbon. And same here as well. That is why you have a phosphate now here in the 6 carbon of the glucose. So then that is -1, negative, because we used an ATP here. So in step 7, to convert the 2 pieces of 1,3-biphosphoglycerate to 3-phosphoglycerate. Again, that is 2 pieces because you have your glyceraldehyde from step 4 and step 5. Okay, so that is +2 here. Then step 10, we converted 2 pieces of phosphenol pyruvate into 2 pieces of pyruvate. We produced an energy from that. We converted the ADP into ATP. Same with number 7. So that is +2 because we produced an energy that is positive. So if you will compute that, the total of ATP that has been produced in 1 piece of glucose is 2 ATP. And then next, let us discuss. So on the previous video, we have tackled the monosaccharides that enter the bloodstream through villi. Okay, right? So the glucose will enter the cytosol of the cell, and the fructose and galactose will proceed on the liver for them to be converted into glucose 1-phosphate. So let us discuss how will they enter the glycolysis, since glyco means sweet and fructose and galactose are all sugars, which means they are all sweet. So where are the entry points in glycolysis? So here is the glycolysis pattern for glucose. The galactose here from the liver, okay, before it enters the cytosol, it will be converted into glucose 1-phosphate, and this glucose 1-phosphate will be isomerized into glucose 6-phosphate. So therefore, the entry for galactose is at glucose 6-phosphate. And then for the fructose will be converted into fructose 1-phosphate, and the fructose 1-phosphate will be cleaved, okay, will be splitted into dihydroxyacetone and glyceraldehyde here. So the entry point for fructose is here from the cleavage, okay, from the dihydroxyacetone here, the conversion, and to the formation of glyceraldehyde 3-phosphate. So that is the entry point for galactose and fructose once it entered the bloodstream through the villi. So that is all for the glycolysis.